Cheers to immune milk!
This is the IGA team, and the aim of our research is to develop value-added milk products that are based around hyperimmune milk. So an immune milk has targeted antibodies in it. So what happens is if you vaccinate a cow against human disease pathogens, then they'll develop antibodies against those pathogens, and that's transferred in through to the milk. And once that milk's collected, we process it through, well, the idea is to process it through to a powder, and then it gets incorporated into food products like yogurt or drinks, or into health products like chewing gums or mouthwash.
In our group, we have developed a patent immunisation regime that induces cows to make IgA-type antibodies. We vaccinate the cow in 3 different sites: the neck, peritoneal and udder. This stimulates the mucosal immune system to produce an enhanced IgA response, which is different to other immune milk products that usually contain IgG-type antibodies. The antibody normally found circulating in blood. For immune milk products, IgA is the antibody of choice because it has special characteristics that protects it from enzymatic breakdown, which is important if your antibody is consumed and exposed to the gastrointestinal environment.
Over the last 5 years, we have worked with several industry players in the development of specific IgA products. We have undertaken a number of animal trials ranging from small pilot trials to larger 1,000-cow trials, testing the process at a commercial scale. Within our group, we have a range of expertise that encompasses the whole process from trial and vaccine design, ethical and regulatory approvals, where we need to ensure the milk can still enter the food chain, immunisation of cows, milk sampling, antibody testing and analysis through ELISAs we have developed.
The milk collected from the cows is analysed by ELISA to look for the high responding animals. That is, the animals that have produced higher levels of specific IgA antibodies to the vaccination. Here we can see quite easily when an animal has a good response, as the darker the colour in the ELISA, the greater the antibody concentration.
So once we've been able to identify the high responding cows, we can collect their milk and process it into products. At this stage, products are normally milk powder. This milk powder can then be tested for efficacy, which simply means how well it works. An example of this efficacy testing is, for example, seeing if the specific IgA antibodies can inhibit the adherence of the bacteria, basically meaning, can we stop these bugs from growing where we don't want them to?
This research will have a positive impact on the Waikato region and further afield in many different ways. Firstly, this research will create opportunities for dairy farmers to improve their profitability. The technology will allow them to develop specialised herds producing high-value milk. And there will be an environmental spin-off effect from this, with higher returns to farmers without increasing animal numbers. This will meet growing demands to reduce our on-farm impact on the environment. The products themselves will create health benefits for the consumer. And last but not least, the research will increase export sales, providing economic returns for both the Waikato community and New Zealand.